Hello and welcome back to part 2 of our genetic technology little mini unit. We're going to dive into a lot of the tools that we use nowadays, currently, for genetic technology, but we're really going to focus in on some of the uses of those tools. So let's get started. To begin with, we're going to do a very quick overview, review of DNA replication, because most of the things that we're talking about today deal with stopping the replication process, messing with the replication process. So let's just review this so that we're comfortable moving forward. So when you start with DNA replication, you have a template strand. That's your original, old parent strand, right? That's a template strand. That template strand is separated with the help of helicase, an enzyme. Once helicase separates the template strand, DNA polymerase comes in and adds new nucleotide bases to match up to that template strand. From there, two identical strands are formed, right? This is what semi-conservative means, right? We have two template strands that have been separated, and then they are half old, half new on these new identical strands that are formed. These new strands that are formed are half of the template strand and half of the new nucleotide bases that were added from DNA polymerase. Important to remember, half old, half new, semi-conservative replication. So, in this process of replication, there are ways that we can manipulate this process. And one of the ways that we can manipulate the process of DNA replication is using what's called polymerase chain reaction. I don't know anybody who actually calls it polymerase chain reaction. It is almost exclusively referred to as PCR, probably because that name is just too long to say. And what PCR does is you take a sample of DNA, and it can be any amount of DNA. It can be a little bit or, kind of a lot, but in a relatively short amount of time, about 4 to 5 hours, it will go through a process of forcing the DNA to go through DNA replication. And so it makes DNA replicate at such a high speed that you end up with hundreds of thousands of copies of the DNA that you want to be looking at. And it's really interesting because DNA replication doesn't take a very long time to do, but this, using PCR, speeds up the process incredibly. So you end up with thousands of your DNA of interest, so then you can just do different tests on it very quickly. One of the things you can do once you have all of this amplified DNA is run a gel through an electrophoresis machine. Now, an electrophoresis machine is a machine that will then separate DNA molecules by size. Now, you may be able to tell that just from this prefix, electro, that it uses an electric current. And that's absolutely true. So what you do here is you load up DNA. Typically, you've loaded it up different sizes of DNA. You have short strands, long strands, medium-sized strands. But you load up DNA into these little wells, these little punch-outs in the gel. And a gel is what you're thinking of. It's kind of like Jell-O, but a little bit more firm. So you load them up into the gel, and then you run an electric current from the negative side to the positive side, right? You're pushing this electric current from one end of the gel to the other end of the gel. And when you're pushing that electrical current through, you are also pushing the DNA with that electrical current. And so the DNA gets pushed through this gel, and you can kind of see it in the second picture where you have these little bands starting to show up. That's DNA being pushed through the gel. Now, because you have different sizes of DNA, they get pushed to different areas. The bigger the section of DNA, the less it's going to move, right? It's heavier. There's more DNA there. It's harder to push something that is bigger than it is to push something smaller. So the bigger the DNA, the less it moves through the gel. The smaller the DNA, the more it can move because it's lighter, so it gets pushed further down the gel. Once you have finished running this gel, electrophoresis, you can actually read the sequence of DNA because all the DNA fragments are in different locations. So it's easy to see which fragment is the shortest and which is the longest, because they will stop in different positions. This might seem pretty obvious, right? Like, yeah, the larger a piece of DNA is, of course, it's not going to move as far because it's heavier, but this was pretty novel, like pretty brand new in the 70s, because it was developed by this guy. And this guy is named Fred Sanger. And Fred Sanger developed what's known as Sanger sequencing, because when you invent something, you can name it after yourself, which is pretty cool. I would love to name something after myself. Maybe someday. So Sanger sequencing is just a sequencing method for DNA. So it is just a way to determine the order of nucleotide bases. Again, this was developed in the 70s, so it's not like super old, but at the time it was like really impressive because there was no really known way to do this until he came along. So what he did is he modified nucleotide bases and made them like fluorescent, right? Like glow in the dark, like very bright. So he modified these nucleotide bases, and when he added these nucleotide bases into DNA while they were replicating, it actually prevented the DNA from continuing on in the replication process. So these modified nucleotide bases were added, and then they stopped the replication process. And because there were so many DNA strands that he was adding these modified bases into, he got tons of strands that were different lengths, which is great because, as you know, you can add different lengths of DNA into gel electrophoresis. And that's exactly what he did. So he put all of these different strands into a gel electrophoresis machine, and from there he was able to run the gel and then read the sequence of DNA. And the way that he was able to read it is because they were all fluorescently labeled, right? As I mentioned, they like glow in the dark. And so he was able to read the different colors to determine what the DNA sequence was, which is kind of cool and kind of a big deal. And it's actually still used today. Although like we have some better ways to do this, the basis of Sanger sequencing is still used today. So this is just an image of what I was just talking about, just to kind of help you visualize what's happening. So you have this primer, right, that's going through replication, and you are separating the strand of DNA, and you end up with just like a regular strand of DNA ready to be replicated as normal. And you add in the replication stopping nucleotides, one for each letter, right? That makes sense because there are 4 nucleotide bases. So you add in a C, a T, a G, and an A into your replication DNA that's about to be replicated. And as it goes through, it stops the replication depending on when these new modified nucleotides go in. And then you put all of these different lengths of DNA into a gel, and you read the sequence, and that's it. Then you just read the sequence from bottom to top, and you have your DNA sequence. It's pretty cool. It's easy and it's relatively quick. So it's a cool little method to learn about. Now, what else can we use gel electrophoresis for, right? Because like that's cool, but like how often are you trying to like sequence DNA? If you're a scientist, pretty frequently, right? Because you can use that to determine like the sequence of genes in the COVID variants, right? That's important. You can also do it to determine diseases and anything like that. But what we use it like mostly for is like DNA fingerprinting and crime scene investigation, right? CSI. So with this is a gel. This is a gel electrophoresis that has been run. You can see all the DNA bands as they move through it. But a different way that we can chop up the DNA is just with enzymes. So the enzymes that we use, we can cut DNA at specific base sequences. Like if you have a specific sequence you're looking for, like CCCGGG, an enzyme will cut between the C and the G. You can have enzymes cut between like any variety of sequences, like base sequences that you want. This is just one example. And when you cut the sequences at specific areas, different individuals will have different size fragments. Because although our DNA is like 99% the same, like all humans have like 99% identical DNA, that 1% is actually quite large because there are like 3 billion base pairs in a human's DNA, in a human genome. And so even just like 1% difference is quite significant. And so different individuals will have different size fragments. And then when you run them, you can, match up the different size fragments. Identical twins have identical DNA. So that is like one caveat. Like that is not, you can't use it for that. But everyone else who's not an identical twin will have different size DNA fragments. So things we can use that for is like paternity testing, right? You can match a child's DNA to determine who the parents are. Like here, you can see that the child's DNA was run in the middle, and then you just match up the bands. The bands have to come from either mom or dad. And you can see in this first one that all of their bands do match up to these parents, but in the second one, they don't. So you can determine paternity based off of if the bands match up. You can also use this in criminal cases by matching suspect DNA to DNA found at the crime scene, right? So in this example, we have a victim and we have the crime scene, like blood or hair or whatever was found there. And so we know that some of this DNA did come from the victim, but some of it did not. And so then you match up those missing bands that are not from the victim to the suspects, and then you can determine who the suspect, or which suspect is like actually has DNA that was found at the crime scene. And in this case, it was suspect 2, and you can see that because this top band and second band match up, this thick middle band matches up, as do these last 2 bands. Actually, like all of the bands match up, but you can see that those match up, whereas the suspect 1 and 3 do not have all matching bands. And the last thing we're going to talk about today is exploring human genetics and disease. So one of the big things that we did when we were going through sequencing DNA and learning more about DNA replication and all of this is creating what's called the Human Genome Project. And the Human Genome Project is a massive, absolutely massive international study of the entire human genetic material. This was like, it took 12 years, and at this, like many anonymous volunteers donated their DNA to determine every single A, T, C, and G and where it's found in the human genome. And this is how we actually know that 99% of humans, of like your average human, share the same genome. That's a super high percentage, and it's crazy that like one-ish percent of DNA that's different accounts for all of the differences that you see in people all over the world. So it's incredible how much DNA we have in common, and yet how much is related to that 1% that we don't have in common. So the Human Genome Project was incredible. It totally changed the way we look at diseases and what the proteins do. So this led to current research, future research. We are continuously striving to improve our knowledge of the genes that are in our DNA, and so we are looking for what proteins do these genes code for, what are the functions of those proteins, what happens if a gene is missing or mutated. Great question. What does it cause? Does it cause diseases? Does it do nothing? Who knows? Is it beneficial to actually be missing a gene or have a mutated gene? All of these things are constantly being asked and constantly being answered. And it's an incredible field of study, and I urge you to look further into this because it is, to me, one of the most exciting parts of science. So I know this was a bit long, but I'm really proud of you for sticking through, and I'll see you in class.